Disc 4.
Berg had come to Stanford as a junior professor in 1959, as part of the package deal that also brought the more eminent Arthur Kornberg there from Washington University in St. Louis. In fact, Berg's connections to Kornberg can be traced all the way back to their common birthplace of Brooklyn, New York, where each in his time was to pass through the same high school science club run by a Miss Sophie Wolf. Berg recalled, "She made science fun. She made us share ideas." It was an understatement, really. Miss Wolf's science club at Abraham Lincoln High School would produce 3 Nobel laureates: Kornberg, 1959, Berg, 1980, and the crystallographer Jerome Karl, 1985, all of whom have paid tribute to her influence. While Cohen and Boyer, and by now others, were ironing out the details of how to cut and paste DNA molecules, Berg planned a truly bold experiment. He would see whether SV40, implanted with a piece of DNA not its own, could be made to transport that foreign gene into an animal cell. For convenience, he would use as the source of his own SV40 DNA a readily available bacterial virus, a bacteriophage. The aim was to see whether a composite molecule consisting of SV40 DNA and the bacteriophage DNA could successfully invade an animal cell. If it could, as Berg hoped, then the possibility existed that he could ultimately use the system to insert useful genes into human cells. At Cold Spring Harbor Laboratory in the summer of 1971, a graduate student of Berg's gave a presentation explaining the planned experiment. One scientist in the audience was alarmed enough to phone Berg straight away. What if, he asked, things happened to work in reverse? In other words, what if the SV40 virus, rather than taking up the viral DNA and then inserting it into the animal cell, was itself manipulated by the bacteriophage DNA, which might cause the SV40 DNA to be inserted into, say, an E. coli bacterial cell? Well, it was not an unrealistic scenario. After all, that is precisely what many bacteriophages are programmed to do, to insert their DNA into bacterial cells. Since E. coli is both ubiquitous and intimately associated with humans, as the major component of our gut flora, Berg's well-meaning experiment might well result in dangerous colonies of E. coli carrying SV40 monkey virus, a potential cancer agent. Berg heeded his colleagues' misgivings, though he did not share them. He decided to postpone the experiments until more could be learned about SV40's potential to cause human cancer. Biohazard anxieties followed hard on the heels of the news of Boyer and Cohen's success with their recombinant DNA procedures. At a scientific conference on nucleic acids in New Hampshire in the summer of 1973, a majority voted to petition the National Academy of Sciences to investigate without delay the dangers of the new technology. A year later, a committee appointed by the National Academy and chaired by Paul Berg published its conclusion in a letter to the journal Science. I myself signed the letter, as did many of the others, including Cohen and Boyer, who were most active in the relevant research. In what has since come to be known as the Moratorium Letter, we called upon scientists throughout the world to suspend voluntarily all recombinant studies until the potential hazards of such recombinant DNA molecules have been better evaluated or until adequate methods are developed for preventing their spread. An important element of this statement was the admission that our concern is based on judgments of potential rather than demonstrated risk, since there are few experimental data on the hazards of such DNA molecules. All too soon, however, I found myself feeling deeply frustrated and regretful of my involvement in the Moratorium Letter. Molecular cloning had the obvious potential to do a fantastic amount of good in the world. But now, having worked so hard and arrived at the brink of a biological revolution, here we were conspiring to draw back. It was a confusing moment. As Michael Rogers wrote in his 1975 report on the subject for Rolling Stone, the molecular biologists had clearly reached the edge of an experimental precipice that may ultimately prove equal to that faced by nuclear physicists in the years prior to the atom bomb. Were we being prudent or chicken-hearted? I couldn't quite tell yet, but I was beginning to feel it was the latter. The Pandora's Box Congress, that's how Rogers described the February 1975 meeting of 140 scientists from around the world at the Asilomar Conference in Pacific Grove, California. The agenda was to determine once and for all whether recombinant DNA really held more peril than promise. Should the moratorium be permanent? Should we press ahead regardless of potential risk, or wait for the development of certain safeguards? As chair of the organizing committee, Paul Berg was also nominal head of the conference, and so had the almost impossible task of drafting a consensus statement by the end of the meeting. The press was there, scratching its collective head as scientists bandied about the latest jargon. The lawyers were there too, just to remind us that there were also legal issues to be addressed. For example, would I, as head of a lab doing recombinant research, be liable if a technician of mine developed cancer? As to the scientists, they were by nature and training averse to hazarding predictions in the absence of knowledge. They rightly suspected that it would be impossible to reach a unanimous decision. Perhaps Berg was equally doubtful. In any case, he opted for freedom of expression over firm leadership from the chair. The resulting debate was therefore something of a free-for-all, with the proceedings not infrequently derailed by some speaker intent only on rambling irrelevantly and at length about the important work going on in his or her lab. Opinions ranged wildly from the timid, "Prolong the moratorium," to the gung-ho, "The moratorium be damned, let's get on with the science." I was definitely on the latter end of the spectrum. I now felt that it was more irresponsible to defer research on the basis of unknown and unquantified dangers. There were desperately sick people out there, people with cancer or cystic fibrosis. What gave us the right to deny them perhaps their only hope? Sidney Brenner, then based in the United Kingdom at Cambridge, offered one of the very few pieces of relevant data. He had collected colonies of the E. coli strain known as K12, the favorite bacterial workhorse for this kind of molecular cloning research. Particular rare strains of E. coli occasionally cause outbreaks of food poisoning. But in fact, the vast majority of E. coli strains are harmless, and Brenner assumed that K12 was no exception. What interested him was not his own health, but K12's. Could it survive outside the laboratory? He stirred the microbes into a glass of milk. They were rather unpalatable, served up straight, and went on to quaff the vile mixture. He monitored what came out the other end to see whether any K12 cells had managed to colonize his intestine. His finding was negative, suggesting that K12, despite thriving in a petri dish, was not viable in the natural world. Still others questioned the inference. Even if the K12 bacteria were themselves unable to survive, this was no proof they could not exchange plasmids or other genetic information with strains that could live perfectly well in our guts. Thus, genetically engineered genes could still enter the population of intestine-dwelling bacteria. Brenner then championed the idea that we should develop a K12 strain that was without question incapable of living outside the laboratory. We could do this by a genetic alteration that would ensure the strain could grow only when supplied with specialized nutrients. And of course, we would specify a set of nutrients that would never be available in the natural world. The full complement of nutrients would occur together only in the lab. A K12 thus modified would be a safe bacterium, viable in our controlled research setting, but doomed in the real world. With Brenner's urging, this middle ground proposal carried the day. Now, there was plenty of grumbling from both extremes, of course, but the conference ended with coherent recommendations allowing research to continue on disabled, non-disease-causing bacteria, and mandating expensive containment facilities for work involving the DNA of mammals. These recommendations would form the basis for a set of guidelines issued a year later by the National Institute